Now I'd like to share with you a little bit about what goes into primary research literature. And I'm going to focus here on the word primary. That means experiments that scientists conduct and their results and their interpretation, not some other forms of scientific literature. And I'm also focusing here on biomedical research literature, in particular life science research, biology and affiliated fields. So the main focus of this presentation is on what's called the IMRaD structure. And IMRaD here stands for some of the components, but not all of the components of typical literature. So on the right here, we can see the cover page of a paper that my laboratory produced a while ago that starts with a title, mitonuclear epistasis, impact, so forth, list of authors, and then there's an abstract, which every paper should have. And that's not listed in this acronym IMRaD. IMRaD stands for Introduction, Materials and Methods, Results, and Discussion. Those are the main sections of research literature. Again, ignoring the abstract, which is always there, but maybe not formally considered part of the structure of the paper itself. Okay, the other thing that's not mentioned that's a common component of research literature is a literature cited or references section, which comes at the bottom of the research paper. So let's take a look at what goes in each of these sections. The abstract is short, as you just saw. It's typically a miniature version, about a paragraph in length, of the entire paper. So it should tell you all of the things that are shown here. What's the big question? What's unknown? What are we doing? What are the authors doing differently? So what's novel about the research? What's exciting about the research? What's their hypothesis or goal? What approaches did they use? And then what were their main findings and their conclusion? For example, was the hypothesis supported or not, or was their goal achieved? And sometimes then future-looking sentence or two at the bottom of the abstract about what might come next. Okay, so abstract, miniature version, it should contain these contents. And this is a great place to look for the summary of what the paper is about. And this is meant to help you decide whether you want to read the paper itself, or maybe find a different research article that maybe is more relevant to what you're looking for. So then we've got the introduction, which is a long section usually that should, although it depends on the format of the paper, some of the short format papers that are like 4 pages in length total to contain all of this information, they don't have the space to give us what an introduction really should do, which is to give us all of the information and details that we need as readers to understand the entire rest of the paper. So it should be all the background information on everything that you would need to know. So again, you can take a look at the bullet points here, the sorts of things that are typically found in the introduction. You'll notice that many of them are things that were also in the abstract. And I'll give you a trick here. Typically, the last paragraph of the introduction does something similar to the abstract in that the authors summarize what they're about to tell you in the rest of the sections of the paper. What did they do? What did they find? What does it mean? Okay, materials and methods is maybe the easiest part of a manuscript to write and probably the most boring to read. I often don't even read the materials and methods unless I have questions that I want answered about how exactly the authors conducted the research. And that includes where they got supplies from, sometimes catalog numbers, so that if I wanted to replicate their work, I could order the exact same chemicals, for example. Recipes for solutions, the experimental protocols, of course, that is what exactly the authors did so that somebody could replicate their experiments if they want to. Sometimes people forget that the materials and methods, the methods part of methods, should contain analysis approaches. So did you use a computer program to analyze some data? Before that happened, did you collect data and truncate some decimal points on some numerical values? Did you round numbers? All those sorts of details should be in the materials and methods. Then we have the results, which I think is sometimes the most difficult to write because it's conceptually odd. Typically in biomedical science and life science research, we often collect data as numbers or as images that we then interpret to decide whether a hypothesis is supported or not. The purpose of the results section is for the authors to write written descriptions of the data, which always seems a little bit odd to me in the sense that if it's visual data, it's present in the manuscript as a figure, typically. And if it's numerical data, it's typically present in the manuscript as a table. But the results section is exactly what's written here, and I encourage you to take a look at several results sections of papers to see exactly how this works, how authors write the results in written or word form, even though they're also showing it as figures and tables in data. Okay. Key point here is the second bullet point. The results and discussion are usually kept separate for a purpose. And in the results section, the authors ideally will make no interpretation of their data. In the results section, you shouldn't see authors saying, we think these results mean something, or based on these results, we conclude that. Right? None of that should be in the results. That's what the discussion is for the authors to take the objective data, images, numbers, whatever the data are, and now is their chance to say what they think the data mean. Do the data support their hypothesis or not? And then the authors have to put their data in context. And this is what's difficult, especially for students sometimes, to write lab reports that have a discussion section or a manuscript. That if you don't have a long history in the field, it's hard, if you haven't read a lot of the literature in your field, it's hard to put your research project in context of what else is known about the same question. So the questions that sometimes are addressed in the discussion are things like, are the data you found, do they support previous research? Or are there times when what you've discovered actually goes against some conclusions that previous researchers made? That's one of the parts of the discussion section, is for you to explain why those sorts of things might be happening. Okay, so that's typically the end of the paper. The authors often end with a paragraph or two, again, about future-looking statements. So what does what they just told you about, what does their research mean for the future? What new types of questions are able to be asked? What was the value of the research that they did? And then finally, literature cited, which is the details that you will use, and I'll talk about this in a future video, how you can find all of the papers, scientific manuscripts that the authors used and cited in their manuscript. Okay. Typically the last section of a manuscript. Now, why the IMRaD structure? In particular, why do you keep the results and the discussion, the objective description of the data and the subjective interpretation of the data, separate? In theory, the idea is that it helps the reader not have their judgment clouded by the author's opinions of their data. That is, we are all, as scientists, supposed to be evaluating the methods and the results and doing our own objective and critical thinking of, did the authors do everything right, essentially? Were the experiments done properly? Did they use large enough replicates? Did they use appropriate control experiments? People have a temptation to, if we just see the data and then immediately see the author's interpretation of the data, excuse me, well, of course, it's easy to go from one to the other without doing that intermediate step of critical thinking. So this is why often the results are presented first in the manuscript. That gives you a chance to interpret the data on your own, think critically about it before you proceed to read what the authors thought their data meant. But in some journals, the results and the discussion are combined together into a single section, which frankly, I think is easier to write as a scientist, because as soon as you say, these are the results from this experiment, you can say right after that what you think that means. And that often helps because multiple experiments are typically presented in a manuscript. And the logical flow of, we did this experiment first and we got this result, and that led us to think about doing the next experiment and we got that result. It's a linear flow, not always linear, but it's presented as a linear flow of logic. And so it's sometimes difficult to write the results all by themselves and then to have to write the discussion separately. Okay. One other point, materials and methods are not necessarily after the introduction. Sometimes they're at the end of the manuscript as well. So again, I just mentioned that the materials and methods sometimes go last. Sometimes the introduction is actually called the background. It depends on the journal. and sometimes literature cited is called the references section. So there are minor variations in the IMRaD structure, but this is very typical for a life sciences biomedical journal structure. Okay. Some other things that are typically found in research papers are not part of that major structure because these are sort of minor elements. First, as you saw at the start of the presentation, there'll be a list of authors. Somewhere in the paper, there's typically a list of the contributions of authors. This is a relatively new thing, maybe in the 2000s and beyond, not in older literature from the 1900s, for example, where there will be an explicit list of what each of the authors on the paper did in contribution to that research project. So did they design experiments? Did they conduct experiments? Did they collect data? Did they analyze data? Did they interpret data? Did they help write the paper? Those sorts of things. The affiliations of the authors, so what research institutions or companies or universities, for example, the authors are from. An acknowledgement section typically is the authors thanking people that didn't do as much to support the manuscript to allow them to be authors, but still people that they want to thank. That also typically accompanies a funding statement. So funding bodies that pay the scientists to buy the supplies and equipment and so forth to do research expect to see their agency, like the National Institutes of Health, for example, or the National Science Foundation, they want to see that you are representing that you got money from them so that they can show their value to the taxpayers. Along with that, there's also often, again, more recently in the 2000s, for example, conflict of interest statement. If any of the authors, for example, work for a company that maybe the authors bought supplies from, that could be a conflict of interest that could cause one of those authors to make unethical decisions about parts of the manuscript. And so it's important for us to know, do any of the authors have a potential conflict of interest? Another recent addition to a lot of journal articles you should see are statements about data availability, or sometimes called resource sharing, which involves the authors making statements about where you can find all of the data, the full data set. Sometimes those are deposited in online databases, so they're not printed as part of the PDF or the print journal article. So they tell you where you can find the full data. And if they've provided, or if they've generated, reagents, new strains of organisms, new chemicals, things like that, they should tell you how you can request those from them. And finally, supplemental files. So everything that doesn't fit in the IMRaD part of a research article, sometimes there are huge data sets, large tables that are important to share with the readers, but aren't critical to the interpretation and understanding of the core message of the paper. Then authors will post those alongside the manuscript online, so you could download extra document files and spreadsheet files and image data sometimes to get the complete data set if you wanted to, for example, replicate some of their analyses. So what I'm going to do now is briefly show you 3 different manuscripts, and I want to highlight the fact that you can find some of the key information you might want to know, like the hypothesis, what species and organism was used in an experiment, those sorts of things, in manuscripts from the journals Genetics, Nature, and Current Biology. So here's a paper that I was an author on from 2004. This is the Current Biology format. Again, a 6-page manuscript, typically. We've got the title, we've got the list of authors and their affiliations, the organizations that they are associated with. And in this case, instead of an abstract, we have the summary, which in Current Biology is structured into a background section and a results section, and then a conclusion section in the second column here. And so let's take a quick look at what's in the summary, which is essentially an abstract. So the background is that brief portion that's introduction to the topic. And typically at the end of that, there will be a statement about the goal or the hypothesis. So we've got gap in knowledge, and we can look for things like previous, the word previous, right? The authors are telling us something happened in the past. Previous studies suggest that something. Here we investigate that thing. So this is a way to find what's the question, what's the hypothesis, what's the goal, what's the gap in knowledge? That's the previous studies suggested something is true, but nobody's looked at it yet. So here we did that thing. And then a brief summary of the results in the results section, and in the conclusion section, the interpretation. Our data suggest that something, and then a future-looking paragraph. Further analysis should do this. So these are the key things, right? We've looked at what's the hypothesis or goal, the gap in knowledge, the approach, summary of the results, conclusion, what the authors, what we think the data mean, and what might come next. All right, so now let's take a look. Again, that's the abstract. Now, the introduction contains all the background information, context that we should need to know to understand the paper. And at the end of the introduction, typically, we will find a summary of the paper, again, similar to the abstract. So here, we take advantage of new tools. So there's an innovation. What's new? What's different? What did we bring to the table, so to speak? 2, here's the goal. Map the genetic basis of something, clone something, analyze something, and study something. Right? So here again, we find a couple of important pieces of information. You can always or almost always look for these things in these spots, in the abstract. And in the last paragraph of the introduction, usually another restatement about gap in knowledge, goal, the approach sometimes, and sometimes, again, an introduction to what the result was. So here we have a combined results and discussion section, so data mixed with the interpretation of the data. Okay, and then in the current biology format, I'm going through all of the different sections of the results and discussion, different data figures. In the current biology format, after the combined results and discussion, then there's a conclusions section. So this is one other place. Typically, this would be in the discussion section of an IMRaD structured paper. Here we have a conclusion section that once again will restate the conclusion of the paper, the goal. These studies show that a single major chromosome region controls sex determination, and then some other keywords and phrases that you can look for things like what should happen next in this line of research. Further extension should make it possible to do something, and this species provides a unique opportunity to do some additional future work. So you can look for these little types of wording that will give you hints to what's the novelty, what's new that the authors did for the first time, why is an organism potentially, or system, research system, useful for the type of research that's being conducted. Those can often be found in the introduction, the abstract, especially the last paragraph of the abstract, and often the last paragraph or so of the discussion or conclusions section. Now, a couple other things I'll point out now. We have the materials and methods, or experimental procedures. So in this case, that comes after the results and discussion section. So this is everything someone would need to be able to replicate what we did in theory. And then let me highlight this supplemental data. I mentioned supplemental data just now, and this is the link to getting all the additional files that aren't part of this 9-page PDF that are additional data that are useful, that are associated with this project. And then one other thing. So we've got an acknowledgments section. That's not the one other thing. We've got the acknowledgments section that talks about funding and thanking people for things that weren't within the scope of each of these people being authors. We've got the references section. So these are all of the citations in this paper. And finally, this is sort of akin to resource sharing data management accession numbers. Aside from those supplemental files with additional data, we also had DNA sequences that were generated as part of this project, and those sequences are available through these resources at GenBank. So that's very much like a data availability section. So all of those things are part of the structure of this current biology paper. So now let's take a look at a longer format paper, one from genetics, 2008, a paper that I was the first author on with my PhD advisor, published in the journal Genetics. This is a longer format. It's a 10-page paper. And similar thing. So I'm not going to elaborate on this too much, but here, once again, 1-paragraph PDF that talks about the technique, for example, that we used and what the data demonstrate. So an interpretation of the data. We've got the introduction, although it doesn't say introduction at the start here, but introduction section. We've got, again, looking for phrases that might clue you into finding things like, what's the gap in knowledge? What's unknown? What did previous research set us up to do here? Our previous analysis suggested something. These findings raised the possibility that something new that we can work on. And so in the present study, that's what we did. And again, like the abstract, a summary of our findings right there in the last paragraph before the materials and methods section. And so we've got the results, we've got some data figures. And once again, I want to point out that let's take a look at the discussion section. Sometimes the first paragraph in the discussion section is much like the last paragraph of the introduction, another place to commonly look for a quick summary of the paper. So in this case, in this report, we used, this is a generalization, we used this technique called FISH to study this question, to compare something, and what we found. Okay, implying that interpretation. So here we've got introduction, gap in knowledge, result quickly, and conclusion, all in the first paragraph of the discussion section. And then if we take a look again at the last paragraph of the discussion, let's see, after some more data figures, hopefully you're not getting motion sick. We've got information about benefit. So comparative studies within this group of fish species, like what's it going to do for us in the future? Promises to yield insights into this thing, right? So once again, sort of what this system is good for and what we can do next is present here in this last paragraph of the discussion. So again, trying to reinforce the idea that you can always find, or almost always find, similar types of content in particular spots of the paper. The abstract, the last paragraph of the introduction, the first paragraph of the discussion, and the last paragraph of the discussion are very stereotypical, usually, about the sorts of content. And they often contain some of those really key pieces of information you might be searching for quickly the first time you look at a manuscript and are trying to decide if you want to read it in more detail. And finally, let's turn to a Nature paper. So this is one of the short format articles, 5 pages in total. In this article, also of which I'm an author, related to the same set of research projects that were in the previous 2 articles we just looked at. So here we've got this typical 4 or 5 page Nature paper. We've got the authors underneath the title. We've got an abstract that contains all of that information that we typically find in an abstract. So here we show that summary of the results, and our data indicate that some big picture interpretation of the data from the authors. Now, in this case, there is no introduction section. It's sort of a combined introduction and results. So the first paragraphs here are basically the introduction. And so if we take a look in that last paragraph, it's again got that typical, here we demonstrate that there is so what's the result? Here's some data with some numbers in it. So a date range for something that we interpret. So typical content here in the introduction. And then a couple specific points to note here. So I'm going to skip through most of this again because it doesn't have a typical results and discussion separate. There's the discussion. So it's had introduction and results together. We've got a discussion section. And excuse me. What I want to point out first is there's a limited methods typically in Nature papers. So you'll see the methods is a methods summary. And you'll want to note, if you ever read a science paper or a nature paper, these really short format papers, a lot of the details, it's a summary because there are additional details presented as supplementary information. The purpose of these short articles is to keep them short so that they're quick reads. And part of that is to not have the methods be extensive as part of the manuscript. So they're often provided as supplemental figures. Okay, so we've got the citations here, the references. We're going to take a look at what's beneath them. Okay, so we've got supplementary information again, so a link to where we can find some of that additional information we might want, especially probably more methods. In the acknowledgments section, we've got the list again of people that we thank for having helped, but each of these people didn't contribute enough individually to become authors, so they're just acknowledged. Here we see typical citations for funding sources, so listing the funding source, the agency, and also referencing the specific grant numbers that gave funding to different authors, these individuals whose initials are in parentheses. And we've got here a key point is the author contribution section. I mentioned this previously. So this is how you find out which authors were responsible for what parts of the manuscript. So these 3 authors conceived and designed the study. Some others contributed reagents, tools, resources, and carried out genotyping experiments and so forth. And finally, again, we've got author information, which indicates that there's data that's available sort of as supplemental information at this database.